Hello student, good afternoon. Today we will teach you fate map in different groups of animals. Fate map in different groups of animals in the groups of animals. Fate map in mollusk. First is fate map in mollusk. Wilson scientists have described in 1904 performed experiments on Dentalian egg. He observed 3 layers of cytoplasm. He observes 3 layers of cytoplasm in the egg of Dentalium, which are as follows. Number 1, a layer of clear cytoplasm at the animal pole of the egg. At the anterior side is animal pole, posterior side is vegetal pole of egg. Second number, a broad layer of granular cytoplasm on the equatorial zone. Equatorial means median. Third number, a clear layer of cytoplasm at the vegetal pole of the egg. Vegetal pole of the egg, posterior side. When the egg begins to cleavage, the clear cytoplasm at the vegetal pole is pushed out in the form of a polar lobe. As the division of the egg draws to its conclusions, the polar lobe is rounded off and remains connected to the rest of eggs by a narrow cytoplasmic bridge. Narrow cytoplasmic bridge. Narrow cytoplasmic bridge. When the division is completed, when the division is completed, the polar lobe is found to be connected. Polar lobe is found to be connected with one of the daughter cells, namely the cells CD, and at the end of mitosis it is fused with this cell. In this way, the cell CD gets all the 3 types of cytoplasm, while the other daughter cells, cell AB gets a half of the clear cytoplasm, a half of the granular cytoplasm at the animal pole, but no vegetal cytoplasm. Second is fate map of protochordate. First is fate map in mollusk. Second is fate map of protochordates. In the blastula of Amphioxus, the blastoderm forms a single layer around the blastocele. The cells from vegetal pole of the egg will become the flower of the blastocele or hypoblast and are the future endoderm of the embryo. Those cells from the region of the animal pole will form the roof over the hypoblast, the epiblast, and will give rise to the ectoderm, that is epidermal and neural cells, and chorda mesoderm, notochordal and mesodermal cells. Most of the cells of the composite epiblast will give rise to epidermal ectoderm. Some small epiblast cells with special staining affinities located at the margins of the prospective endoderm. layer form a ring around the blastula show that this ring of cells consists of two general areas, the dorsal crescent and ventral crescent. The cells of the dorsal crescent give rise to the presumptive notochord and neural plate cells. The ventral crescent contains the cells that make up presumptive mesoderm. Third number, fate map of cyclostomes and fishes. Fate map of cyclostomes and fishes. Fate map of cyclostomes and fishes. Nowadays, gastrulation is regarded as a coordinate series of folding and movements by which the various groups of the cells are arranged id three main layers: ectoderm, mesoderm and endoderm. 3 layers. These cells of the specific movement in different directions form the different areas, while are termed as the presumptive areas. They are arranged in a definite fashion and brought a natural relation with another. It is clear that such a fashion of arrangement starts from a type of egg which is relatively small and have complete cleavage. The cyclostome and primitive fishes both have the map closely related to one another. The blastopore is placed low down of the vegetal pole with an area of endoderm which extends right round the egg from the dorsal to ventral side. Within this ring, presumptive axial mesoderm is concentrated towards the dorsal side and extended much further lateral in the blastula. After gastrulation, the upper part of the egg is occupied by presumptive endoderm. The dorsal region, which will become the neural plate and remainder into epidermis. A very similar method is found in the protochordates. Fourth number is fate map in amphibians. Fate map in amphibians. The blastula of the amphibians embryo is also a round structure that can be roughly divided into three regions. They are similar but not identical to those of Amphioxus. Number 1, the vegetal region. It includes hypoblast, which comprises yolk filled pigments less macromeres. It represents presumptive endoderm, which contains material for the formation of midgut and hindgut of the embryo. Second number is the animal region. First is the vegetal region. fate map in the amphibia. First is vegetal region and second is the animal region. Animal region. It represents epiblast which comprises micromeres. Unlike the blastula of Amphioxus, the epiblast of the amphibian is several cells thick. In the frog, the darkly pigmented micromeres derived from the animal pole of the egg gives rise to the future ectoderm of the animal. It includes two main regions: region of prospective epidermal ectoderm which develops into the epidermis of the skin of the embryo. Region of prospective central nervous system. Second number is region of the prospective central nervous system which contains material for the brain, spinal cord and sub areas for the sense organs such as eyes, ears and nose. Third number, the intermediate or marginal regions. The third area is the gray crescent area. Gray crescent area, which in blastula represents in ring of the cells around the equator. Most of these cells are the presumptive mesodermal cells. Unlike the blastula of Amphioxus, in which the presumptive mesoderm cells occupies approximately half of this marginal zone. Ventral crescent, the presumptive mesodermal cells of the amphibians almost encircle the blastula. It contains the following subregions: notochord region, which is present on the dorsal side of blastula and give rise to notochord. Area for anterior part of alimentary canal, that is the situated below the notochord, below the notochordal area, and gives rise to endodermal lining of the mouth, gill region and pharynx. Region of the somites or segmental muscles, that is present on the both sides of the notochordal area and develop into the trunk muscles or somites, and ventrolateral mesoderm area, which lies on the lateral and ventral parts of the marginal zone and gives rise to the mesodermal lining of the body cavity. The kidney and reproductive organs develop. Last is fate map in birds. Last but one, fate map in birds. The blastula of the birds consists of two areas, area pellucida and area opaca. The area opaca is completely excluded from the formation of the body of the embryo. The fate map is therefore restricted to the area pellucida only. Further, since the blastoderm is bilateral and consists of epiblast and hypoblast, fate maps have to be made out for each of the two layers separately. Last is fate map in mammals. It is impossible to mark or stain the cells of the mammalian embryo and follow their path through the sequence of developmental phases. The rat, mouse, rabbit, pigs, and monkeys are the eutherian mammals most studied, and much of our knowledge is based on these forms. Despite the fact that the mammalian egg undergoes holoblastic cleavage, the cells composing the inner cell mass spread out and form a plate of disc resembling the blastodisc of the birds and reptiles. The mammalian blastodisc too become divided into epiblast and hypoblast as in birds and reptiles, but there is no consensus as to how this occurs. Regarding of its origin, the hypoblast will give rise to the endoderm as in other animals. The cells remain in the inner cell mass are considered to be the epiblast and will give rise to the epithelium, neural ectoderm, notoderm, and mesodermal components of the embryo. Thus, the mammalian fate map resembles that of the reptile and birds. Okay, thank you.